So just, I guess you know from our conversation before, I'm a computer scientist. I have modeling projects, and so I've chosen a few, excuse me, a few modeling projects to talk about this morning. I had three things I thought I would be really interested to talk about, and I'm happy to, wherever you'd like to take this. But one is this concept of mapping diverse intelligence spaces and what do particularly developmental spaces look like. And one of the questions that's come up as I listen to your podcast is asking, when we try and unify something like the electromagnetic spectrum, we end up with a spectrum or a periodic table. But when we try and think about developmental spaces, what is it we're looking for? And so I guess the approach I take in my work is that it's intrinsically, you can think of it as tripartite. There's what in your work you've been calling the Platonic spaces or the Platonic function. What's the function independent of how it's implemented? And then a computational formalism or an algorithm or a computer program. And then you've got something that you might consider the mechanism, the physical trace, or the device. And I think thinking of these three, particularly in areas of robotics and biology, is really useful for a computational modeler. I sit for my stance in the middle at this computational formalism. And then in the second and third parts, I thought I'd briefly touch on memory and memory phenomena. And I'm intrigued by this idea of how a basal memory trace can have a sense-making goal, which comes from the TAME approach. So I'm hoping we'll be able to discuss some of that. Okay, so starting with the first part, trying to think about how we map an ontogenetic space. What does that mean? So what is development from a computational perspective? So this is an old project with three quite remarkable PhD students, Nick Gibb, James Watson, and Kai Willitson. And we were looking at how you'd go from a DNA sequence. At the time, we were thinking of them as the genome. Now we'd be thinking about the expression of that being the bioelectric expression, gene expression, bioelectric expression, in the network interactions as this intermediate representation. And then James was looking at L systems to grow plants, and Nick was looking at lineage trees. And I thought I'd pull something out of Nick's work. So obviously you're familiar with C. elegans on ontogeny. So thinking about a cell, the zygote divides and then divides again, and you end up with an organism. So Nick was taking this and trying to say, how could you get a network model to reproduce these lineages? Not so much because you want to understand C. elegans in this particular case, as because that process forces you, it's a very disciplined way of asking what is a developmental lineage, what do they look like, and then what do these gene expression networks do in terms of creating them? So he started with every cell contains the same set of genes and the same set of gene interactions, but the expression is unique to that cell. They have inputs, so morphogenetic cell-to-cell signals. And then he evolved that. We were doing a lot of work in evolutionary computation at the time and ended up with lineage trees. It's very hard to see at a glance this whole space. So he created what we called LinMap, which was a way of mapping. So the green colored heat map here is 60,000 lineages, each mapped according to a single measure of their complexity. And he tried 6 different measures of complexity, but this is just one of them. And you can then go in and use that heat map space as a way of indexing what lineage trees look like that generate it. So the large black region is cells that divide and divide again, divide forever. So these are you can think of them either as bacteria or in a multi-celled organism. As we were doing here, it would be cancer. There's no differentiation. In other parts of the space, you'd have these very small, sparse lineages which differentiate and stop dividing very quickly. And then you've got other structures. Some of the structures show this beautiful semi or quasi-irregular patterns where the subtrees are repeated across a lot of the different branches, but not perfectly. And then you get ones that are more complex. And the interesting thing about mapping the entire space is you see all the different complexity classes you'd come to expect in complex system science. So you see regions where there's just a single attractor, nothing much happens. You can perturb the system and you get no difference, through to the most complex regions where you're going to expect computationally complex phenomena. So this is where, if you're mapping the attractors in this space, then the Lyapunovs would be close to zero. So the system itself has some stability. It's not chaotic and it's not collapsing into a single attractor or a group of pre-specified attractors. So then Nick was asking this question, are all phenotypes equally available for natural selection? Because that heat map shows the range of complexities in ontogenetic space, but it's not telling us about the likelihood of a specific lineage. So the traditional view in evolutionary computation is that pretty much any um, any character trait is reachable by a small amount of a random walk or a mutation in that space. So mutation can enable you to reach everything. But of course, one of the things we've learned in evolutionary computation is that there are a lot of different biases in the system built into the structure of the genome. And we were particularly looking at what does the developmental process do? So in a sense, what does the developmental process give you in terms of constraints on how you move through that space? So to examine this question, we went for the simplest possible two-cell network we could think of. So instead of evolving C. elegans, this one is just a random set of what is generated by a network. And at some point, it will differentiate into either A cells or yellow cells and B cells, red cells. This is a computer scientist view, not a biologist view of A cells and B cells. And then you can create a fate distribution map. So if you have 4 red and 4 yellow cells in your final organism, you can then say, well, what happens if you evolve the lineage for one more time step instead of differentiating? And you'll end up with 8 red and 8 yellow. And then if you allow the yellow cells to divide one more time, you might end up with 16 cells. So actually, an organism with 4 red and 4 yellow cells is very, very close in this developmental space to one with 8 red and 16 yellow cells. And it's much, much more difficult in this sort of space to get one that has one more red cell or one less red cell. So you can then map this. So take 10,000 samples and look at the cell fates, and you can try a traditional Markov process or stochastic control process where if you don't have a developmental system, you can say, what are the cell fates? So A and B are randomly assigned to the terminal cells, and you get a cluster around the most frequent organisms you develop in this, in terms of the number of A and B cells, are all clustered together. And the question is, what happens when you then put in the network and try and map it? And something quite remarkable happened where when you have these dynamic gene expression networks, you get something that doesn't look anything like the Gaussian structures you would see if you have a stochastic control process. So what we found is, again, 10,000 networks mapped. The most frequent are in the same place, an equal number of A and B cells. The size of the organism is specified by how many steps until probabilistically all of the cells will differentiate. But the neighbors are in very different places. And we actually called this distribution, we don't have a name for this distribution, but we called it our diamond distribution. And it looks very much like the facets of a diamond. And when you think about this, instead of the neighbors being one more or one less cell, the neighbors are major jumps in what you can think of as high-dimensional hyperspaces, or the corners of Boolean hypercubes. When you project them down onto structures, you get something a bit like this. Now, these are not Boolean spaces, but it's interesting to try and visualize what this actually means in terms of gene expression. So just pausing with this for a moment, what I think such a map shows is what you can call the adjacent possible. So you think of it as the bioelectric neighborhood of ontogenetic space. And the fact that it has this highly structured representation was just fascinating. And I'm wondering whether you found this in any of your work. My hunch is that the facets of the diamond are actually pullback attractors in developmental spaces. I don't have any evidence for that, but I'd be intrigued to be able to discuss it. And then I guess the other thing that I found fascinating here is trying to think about looking for xenobots in these spaces. Are xenobots pullback attractors in frog biology, or are they like, you could think about this diamond distribution as being the most frequent distributions that you observe, then reified by evolutionary processes. And xenobots don't need the reification because they are neighbors in this space. So I guess maybe that's a point to just pause and say, do you have any comments around this? Or shall I just keep going with the rest of it?
Well, keep going. I've got many things to talk about. I'm just taking notes as we go. So please keep going, and then I'll come back to things.
Okay. Yeah, so I guess one other thing about trying to think about what's a xenobot is trying to think about whether large language models are a kind of xenobot for human languages and cultures. And that's something that's fascinated me as well. Okay, so the second part, I was going to think about memory and this idea of towards a unification of memory across multiple scales or multi-scale competency architecture. So I did a postdoc. I'm a PhD in computer science, but I did a postdoc in psychology studying episodic memory structures, in particular tensor, so rank 3 memory systems. And episodic memory is interesting because it tries to encode everything about an event. So who, what, when, why, how. So that's context. And then within a given context, it can associate cues and targets. And the kind of neural networks you would use to model this, or we used to model this from Mike Humphreys, Bain and Pike in 1989 was a paper in Psych Review that set up a lot of this work. And the nice thing about these structures, they're like one input layer, one output layer of a neural network, which everybody who works in this space would realize you can learn orthogonal patterns in a single trial. So they're very efficient, but you can't learn different associations easily uh, without catastrophic interference. But if you add in context, you can learn in context. And the context is not added to the queue, it's a cross product with the other two, which is what gives you the rank 3 tensor. So these are very efficient, very effective, but they only really work when the information is already orthogonal. So it's already represented either in a form that could be symbolic or what they call one-hot encoding. The other episodic structures that I think are really interesting are the ones that model neurogenesis. So in dentate gyrus in the mammalian hippocampus, this is a region that has new brain cells that develop all the way through adult life, including adult humans. As I tell my students, they should go out and do a lot of exercise because it generates new cells in dentate gyrus. And then it's no good just doing a lot of sport. You actually need to do something to cause them to differentiate and integrate. And what they do in this region is they encode novel events and also unexpected rewards. And Brad Imony created a beautiful model of this. And then Lara Rangel in Andrew Chiba's lab actually tested it in rats and showed over different time periods these cells integrate and collect together information over time. So these are episodic memory systems. The other way of thinking about neural networks is the classic multi-layered encoders, and these are much more semantic memory. So when we were doing memory research, we would differentiate between memory and learning. Memory can be done in a single trial. It's not always done in a single trial, but it can be, and learning takes longer. So encoders are actually learning systems if they require learning either a compressed or an expanded representation in the hidden layers. And what this is useful for is when you have non-orthogonal vectors. Then these are feed-forward systems, the traditional multi-layered neural networks. In the when experimenting with these, people like Jeff Ellman and others added recurrent connections. And so if you take a multi-layer system with recurrent connections on the hidden layers, you get a simple recurrent network. And that brought in the idea of learning to predict in a recurrent system. And interestingly, although it's copying back, the context unit just copies back the previous context, it can build up structures and learn semantic and syntactic structures. And Jeff Ellman used it for grammar learning. So the input predicts the next input, not the current input. So it's not like a typical encoder. It's encoding the next step. This is very much like large language models today are based on this idea of predicting the next input with something that is either recurrent or unfolding. These recurrent layers is another way of thinking about them. So it's interesting from a modeling language point of view, because you get both syntax and semantics, but unlike a traditional Chomskyan linguistic approach, these are not distinct and separate, but rather they're integrated within the same representations. So exploring this notion of how an item in memory might have some form of goal or be an active element. I think it's really interesting in studying these simple recurrent networks to think about who is the actor and who is the object. And then in a feedback network, any feedback network doesn't have to be a simple recurrent network, but there's always a duality. There's inputs act as operators on the activations already in the network. Activations in the network act as curried functions over the input sequences. And the connections within the network say what kind of curried functions are possible in that system. So we were playing around with this. This is work by Anthony Bloch and I, and we were looking at how these networks could learn really simple. A three-function task is a task where there's a sequence. You give an AND, an OR, and an XOR as an input pattern, followed by a whole sequence of zeros and ones, and it has to compute that function as a curried function over all the rest of the inputs until it gets another function and gets reset. So we were using this as a way to think about operators and curried functions in neural networks and realized that if we really believed what we were saying, we should be able to map them. So we map the states for each function within the hidden unit space. And so looking at the hidden unit manifold, mapping the first and the third canonical components, we can actually see the geometric structure of how the hidden layer is shaping the data as it passes through it. And then you can map onto that the active patterns. And what an active pattern does is cause transitions in this sort of space. And so you can map, there are 5 different input types, so there are 5 different operators, and you can map the transitions within that entire space for all of those operators. Or you can think about each of the 6 major regions in that space that compute this function as being curried functions, and you can start from positions in those spaces and ask, what does the input do? What does it do to the inputs? So you can look at it in terms of operators and curried functions, and you can actually map these dynamics. And this was all done with Boolean inputs, real valued hidden units. And so then thinking about, again, trying to connect to the TAME framework, one of the questions you can ask is the activations in the neural network are really like the bioelectrics in cells. And we can ask, what would a xenobot be in this space and how would we go looking for it? So there's a couple of different ways we could think about it. One is to relax the idea of the inputs. Instead of having them Boolean, they could be real valued. What would that do within the system? Would the rest of the hidden unit space actually force attractors towards the hidden unit manifold that we see already? Or would there be additional attractors that we could discover in that space? The other way you can think about it is rather than starting from one of the canonical positions, you could start from anywhere in the hidden space and ask what happens. And then, of course, you can combine them with real valued inputs exploring parts of the space. And at the time, we didn't think about how we'd develop those attractors, but I think it's possible now with neural networks to actually look at the attractor dynamics for an entire space, the space that your typical function actually reaches, but then also the rest of the space as well. Okay, so when you think about memory, it's not just thinking about the memory trace, um, and that what that active memory pattern would do, but it's also thinking about what an access process is over that memory trace. So again, going back to Mike Humphrey's work, Mike was my postdoc advisor, modeled with the memory tensor. The memory tensor is constructed by the outer product of a queue and a target and a context. And then you can either use that queue or use a unit vector instead of a queue. And then you can look at, do you actually look for a recognition where you're trying to produce a yes/no answer? Um, uh, do you know your phone number? Yes or no? You don't have to actually recall the phone number to know it. Queued recall is where you have to produce it. So someone says, what's your phone number? And you have to come up with, in a memory system, it would be a full vector, not just a yes/no answer. A familiarity rating or a lexical decision can actually be independent of a context. So it's averaged across all possible contexts or calculated across contexts. And then free association is where you're not giving a specific cue, you're just asking when you think about breakfast, what do you think of? And you might think of coffee. So the conjecture here is that a memory trace could be said to have a goal when it directs energy towards its own encoding, storage, and retrieval, or it doesn't necessarily need initially to be doing all of that. A trace could be just an encoding, which is not, which the organism or the memory trace is not necessarily putting energy into. And then if it is useful, you can imagine an evolutionary process that then encodes it to the point where it becomes critical for the organism and so ends up with energy being put into a particular type of encoding. So just before we leave memory, I've got just two slides here. One is thinking about memory not just as generalizing in terms of access, but also as partitioning. So as I just said, any nonlinearity could actually be a memory trace. It doesn't have to be intentional to start with. But thinking about the active retrieval system, it needs to surface what's relevant in the moment, but also to suppress what's not useful. And so this sets up the notion that partitioning is actually intrinsically useful to memory. A, a lot of people would think of this as being part of a cognitive system, but if you think of it as part of the memory system, um, it gives you very powerful data structures as a computer scientist. Potentially also useful in psychology, though I was looking more at it as a computational device. So just in terms of an example where we did that, um, so we were working with these language learning robots called lingo droids. And in one study, they were exposed to multiple languages. So I don't know if you know the history of language languages in Australia, but prior to European colonization in the Northern Territory, people would typically be speaking 10 languages. You couldn't marry into your own language group, which meant everyone was multilingual. Your parents were multilingual. Your spouse would be multilingual, and your children would have to be multilingual. So with 10 languages, we were working with a language center where they were working with some of these languages. So languages like Aloha and Rembaranga. And we were asking ourselves, how could a robot learn to be multilingual? Because when you think about a 2-year-old child, they're learning language, but they're not told which language they're speaking. They're not told this is Aloha, this is Rembaranga. They just have to learn it. And so one of the things with children, that even today, when you bring up children multilingually, it helps if one person speaks one language. Maybe the grandparents speak Portuguese and the grandparents on the other side might speak a different language. Or one parent might speak one language and one another. So we gave this contextual information to the robot of who they were speaking to. So we included the interlocutor, and it turned out that, and we were using the tensor memory system. So one-shot learning, words could be learned in a single trial. And the robots did learn successfully to be multilingual, in this particular case, two different languages. When it didn't have a word, it would generalize across all the different words it knew. So it would get, a confusion across languages. But if it had a word already with a given person, it would use that word. So it's really interesting that this kind of memory structure enables us to separate language. So you think about the language you speak to your grandparents compared to the language you speak to your mates in school. It's you use it's a similar grammar and it's still English, but actually the word choices can be very different. You can generalize, but you choose to use the words for the people you're speaking with. And so this was a real insight to us in terms of the power of partitioning within the contextual system. And so the last slide here, I thought it's worth touching on the power of this. In consciousness research, people often mention dissociative identity disorder, and it's in the DSM-5, but a different way of thinking about it is that it's not actually intrinsically a mental illness, but rather an adaptive way to live with a history of trauma. And so Maggie Walters is someone who grew up with NPD. She describes herself as a high-functioning multiple. And her alters, she calls her superpowers. And she's written this brilliant memoir where she's written the major part of it, but the alters have actually dictated various chapters. And Maggie credits her alters with literally keeping her alive. And from a memory research point of view, I met Maggie at the Byron Writers Festival several years ago before this came out. And then I read her memoir last year. And when I met her, I didn't realize that she was multiple, and high-functioning multiples typically you will not realize. But trying to think about it from a memory perspective, George Blair West is a psychiatrist who wrote the foreword to Split, Maggie's memoir. And he says straight out that DID is not intrinsically a mental illness. He calls it perhaps the most powerful adaptive response that the human mind is capable of, and it allows a child to hand off the traumatic experiences to an exiled part, and then another executive part can face the outside world, which is what a child facing that kind of trauma needs to be able to do. And one of the reasons I thought I'd include this when we're talking about memory is that one of the approaches that was taken in psychiatry to DID was reintegration treatment, and it has actually caused a lot of trauma to people like Maggie and others. And she does speak about this. She does a lot of public awareness raising. What reintegration does is confusing the trauma with the dissociated identities. So the trauma is what is the problem, and the dissociated identity can be seen as being a way of managing that. So this is thinking about why memory partitioning can actually be seen as really, really powerful. And I think there are perhaps some ways of thinking about that from a basal memory point of view as well. Okay, so moving on, just the last bit, part 3 was just exploring language and communication systems. So Lingodroids was a project that ran for over 10 years. It involved a whole variety of different robots, neuroscientists, language studies, postdocs, and PhD students. And we did a lot of work in simulation and also in physically embodied robots of different kinds. The previous study that I mentioned about the multilingual robots, I should say, was done in simulation, not with real robots. In theory and practice are the same. In practice, they're not. That's a quote that roboticists always used to say when we were trying to move from simulation to real robots. And I think this speaks to the idea that when we say machines are not machines, it highlights the difference between what you might think of as the mathematical space of functions. So this is, you might think of as Platonic spaces, but then the algorithmic space of formalisms, and then the real world of meat and metal bodies. And in the real world of meat and metal bodies, in practice, there is no perfect implementation either of a mathematical function or an algorithmic formalism. Um, and I think this is really interesting when we're trying to explore the real robots, and particularly for language and communication. So with Lingo Droids, I've just got two studies I thought I'd mention here by Scott Heath. Uh, and Scott was interested in a whole range of what can robots learn um, in terms of languages. But he was interested in this one particular question. If humans are going to communicate with robots, can robots communicate with robots? Can they evolve a language when they're heterogeneous? So a lot of the assumptions in cognitive psychology were that the reason why lingodroids, we had this to start with, were so successful is because they have a similar physical body structure, and then slight differences, and then a similar cognitive architecture. And so Scott was saying, well, supposing they don't. So there are different kinds of robot mapping systems. I don't know if you're familiar with this, but a laser scanner will estimate range, and it will create an occupancy grid, a map of its world based on probabilistic filters. So it maps boundaries. It has no concept of time or events. It doesn't store episodic memories at all. The little IRAT robots we were using, they have a forward-facing camera used to estimate appearance. They create these hippocampally inspired topological maps called RatSlam. The version we were using was OpenRatSlam. And they're based around an episodic memory system, which creates networks of events in space and time. So two totally different ways of structuring memory. And Scott was asking this question, can they evolve successful languages for space, for distances and direction? And it turned out that they do. They were remarkably successful, not 100%, probably around 80%. And the lesson from this one is that robots don't need the same body. They don't even need the same cognitive architecture. What they did need, the language games work when the robots are both playing the same game. So one way of thinking about this is they needed the same culture. So the second study I thought I'd mention from the Lingo Droids was with a robot called Opie, which we were using in a child development lab to look at what children can learn from robots and teach to robots. So this was a study where the child was teaching a robot color names. And this is another question in how humans and robots can communicate. So again, it was a language learning study. The robot on its tablet would produce a color patch, might be a blue patch, and the child would name it. So it would say, what color is this? Blue. It might put up another patch, another blue, but maybe lighter. What color is this? It's also blue. Are they different or same? And the child might say, oh, they're different. And then what's the difference? Lighter. And the child would say one's lighter than the other. So in this space, it could pull out the robot could pull out a structured view of both the colors and the relationships between colors. And the interesting one about this is Opie and the child don't sense color in the same way. The child is producing RGB patches of color on its tablet, and yet they're sharing the experience. And furthermore, the child is using vision, it's a sensing. The robot is not even sensing the patch, it's producing it. So these are languages not just for transmitting information, it's actually connecting the private realities of these two very different agents. And they don't even need to be doing the same thing in the same experience. Again, they need to be playing the same game, a language game, but they can be having different parts of the game. And there's still this way of creating this external language that connects their private realities. Last slide, just mentioning current studies. I've been working with Bill Bingley and Alex Haslam over the last 4 years, particularly studying intelligence and looking at groups. And when we study cells and how they work together in biology, often it's a very hierarchical structure. But when you study humans and how we work together in groups, we're always part of many different groups. So this notion of multi-group social intelligence, you're not 100% part of a group and you lose your own individual identity. You retain your identity. And so in this particular work, we were looking at what is a good definition of socially minded intelligence, where an individual can look at their own goals and look at choose a group that they're part of, or the environment might make a group salient, and then looking at the group's goals and working towards group goals or individual goals. For people who are socially minded or have social minded intelligence, you actually recruit other people towards your own goals. And so, how do you look at what we call these aligned abilities for a particular time or task or event? And thinking about deconstructing it into socially minded abilities to recruit resources, and then what resources are available in that context. So, this is just to mention current work. A lot of my current work is in human-centered AI. And one of the practical projects for all of these, we usually have some theory and then also some practical projects. And we've been working on language in the Florence project, which is trying to develop a personal knowledge bank and ecosystem of communication devices, so both hardware and software, that enables people living with dementia to have quality of life at home. Okay, so that's coming to the end of the talk. I will stop here, and I'm open to comments or questions.
Yeah, thank you so much. Amazingly interesting body of work. There's so much here. Okay, a few things I wanted to check into first. So first of all, you mentioned in the robots different kinds of bodies. Have you looked at all at what kind of architectures make it easier to move personalities, specific memories, and so on across bodies with different architectures? I'm thinking of the butterfly caterpillar business, where not only do memories survive the total refactoring of the brain, but they actually get remapped because the butterfly doesn't do things nor care about things the way that the caterpillar does. What have you ever played with that in your system?
We haven't done that explicitly. I've thought a little bit about that, about the butterfly brain case. When would it be useful to do it, and how would you do it? And when you think about different ways of memory encoding, you can encode an entire event, or you can just have the lowest possible or the fewest number of variables. So if you have very few variables, and those variables, so it might be something like a smell. And a question I was going to ask you about the butterfly case is, does the butterfly choose to lay its eggs on those particular leaves that it either favored, or avoid leaves that it disfavored as a caterpillar? So is there actually an evolutionary advantage to the butterfly to retain that memory?
So there's two. Okay, so in that case, there's two sets of things going on. There is definitely an evolutionary component, which is that they don't want to compete with their larval form, and so they try to stay away from each other in some ways. There is that, but that's not the what I'm talking about is a completely different thing, which is it's an experimentally induced memory. So what they did was, and actually one of the people, Doug Blackiston, who did a lot of this work, it was a staff scientist in my group for many years. He just became faculty last month. uh he what they did was, and then there's also some Russian work going back to the 70s for this, where you train the butterfly, you train the caterpillar to see a particular color stimulus, and then to crawl over to that stimulus and eat the leaves that you're given, okay, at this thing. The butterfly now will remember to do that. But what's interesting about the butterfly is that A, it doesn't crawl. So instead of a soft body controller, it now has a hard body, completely different, right? Totally different
Right.
controller. And it doesn't care about leaves at all. It wants nectar. So the reward it's going to get is completely different. And yet, right, you have to keep that new memory. So that's not an evolutionary memory. That's a behavioral, episodic memory that it acquired, and then somehow And we have all kinds of really wild examples of, and so this goes back to something else we're going to talk about who's the actor and who's the object, in the sense that some of these memories have a pretty active component to them. And I suspect, this is like James's thing, where thoughts are thinkers. Yeah. This, like, I suspect that some of these memories have an agentic component to them. They like to occupy new media, new environments that they can live in. Maybe they even do some niche construction to sort of modify wherever they end up to make it easier for them to do their thing. And we have all kinds of weird biological example of this. First of all, planaria, when you train them for a particular task, you can cut off their heads, and then the tail will just sit there doing nothing for 8 or 9 days. Then they regrow a new brain, and wherever that information is, we don't know where it is, wherever it is, it gets imprinted onto the new brain. So it moves from, presumably from the body or from somewhere even weirder, it moves onto this new brain that has to get imprinted from scratch, right? So it's moving, it can move in and out of body tissues. We have other examples where we can cut a little piece of a two-headed flatworm. This is, of course, non-genetic. None of this is genetic. So we cut a piece of two-headed flatworm, stick it into a one-headed host, and something like 17% of the time, that tiny little piece convinces this whole giant host that it's going to be two-headed from then on, you know? So like
Mhm.
the propagation of these memories, and we have other examples where we can put in bioelectric memories, and sometimes they take over, and sometimes they get wiped out depending on how it goes, right? And it's a very important biomedical direction to figure out what patterns are convincing, because we would like to get the buy-in of the cells and tissues when we make our interventions. We don't want to fight the tissue. We want it to accept the set point. So yeah, I mean, it seems like in these cases, like you have a really good model of this potentially.
Yeah, so I think it's interesting to think what gets buy-in and what doesn't.
Mhm.
Or probabilistically, if sometimes it gets buy-in and sometimes it doesn't. So, how do you make the sweet spot, the 17%,
Yeah.
into, 50 or 60 or 70%? That's a really interesting question. In some of the heat maps that we created, we were modeling spiking neurons, and we had a very simplistic model and a network. And Pete Stratton was doing this work, and one of the presented it to an early stage of a grant we were part of. And one of the electrophysiologists stopped him 30 seconds into his talk and said, "You haven't included um, the, um, the most important part, which is after the spike, there's a period at which, everything goes fallow for a bit.
Mm.
And if you don't include that, you haven't modeled it." And he just wouldn't listen to the rest of the talk. So Pete and I sort of, what do you do after that? You're working with groups, you don't understand the electrophys, but he's the expert. So we thought, okay, the first thing we'll do, we didn't think it'd make any difference at all. So the first thing we did is that night, modeled, added this whole component into it. And it turned out the sweet spot went from very, very tiny to this massive region of space, because when you are trying to get a computational um, element, so something that actually can retain its own state for a long period of time, there need to be certain delays that stop the system either jumping immediately to chaos or immediately into one of the attractors. So you don't want a system where the attractors are perfectly defined and the ball will roll down into one of those tractors immediately. You would like it to be able to wend its way through several computational steps before it makes a decision. And so building in um, these sorts of delays in the system was something we started looking for in our networks. So in trying to think about medicine, this is kind of a long bow to think about this as a metaphor, but trying to think about what is the underlying attractor dynamics? What are the lyapunov in that? So how quickly does it diverge to an attractor? How large are those attractors? And then what is the some work that Kai Willitson did in how you map the structure of those attractors and actually visualize them in high dimensional spaces. So are they really fragmented? If you look at something like Saccharomyces cerevisiae, and you have a single cell that's got to deal with the world on the world's terms, in terms of temperature and all sorts of things. It has quite a fragmented structure. It has quite a lot of complexity and a lot of different switches between basins of attraction. Whereas if you look at something like uh, blood cells or cells in and a multi-celled organism where you can be pretty sure the temperature is going to be very stable, the attractors can be much larger, dominated by one main attractor of how it works with just a few switches. And it can be, you can even have more genes in your gene network, but the whole system will be much more stable. So, um, thinking about this in terms of what is the underlying landscape, can you map that landscape? And then where do you place, where is the system within that landscape? Is it deep in the middle of a very stable attractor? Is it right on the edge between them? Are you trying to get it to jump from a stable place to an unstable or an unstable to a stable? What are you trying to do within that attractor space?
Hmm. Yeah. Uh, one we have well, two things come to mind. One is we have this one very strange case where we make these planaria and they're headless, so they have no head. And so they sit there, and typically the way you do these experiments, you wait till regeneration completes. You can tell when it's done, and then whatever the phenotype is, you call it, and then you move on, and that's the end of that. Uh, for whatever reason, we held on to these animals for months, and what we found is that somewhere between 4 and 6 months, they suddenly regrow a head. In other words, they it was fine at the beginning. So the set point was okay as a no head, completed regeneration that served as a stop point. Okay, stable, and then something was still ticking, and there's some crazy, like, I can't think of what biological timekeeper has that weird. It's just like it's too long for anything normal. I mean, I know cicadas do whatever 13 years, but still, like, like 4 months. Like, what process takes 4 months? You need some kind of clock in there. Something is happening, and one interesting recent idea is that what it's doing is it's searching a difficult space for a solution, and that's what takes 4 months. Is that it isn't that there's some clock that's counting down specifically the timing. It's that it's trying to find its way out of a complicated, and usually, I mean, we're usually pretty amazed at how fast they solve these things, but maybe in that case, either because the system itself has been dumbed down to some extent, or the it's been chased into some corner of the space from which it's really hard to extricate yourself, right? That it's maybe it's something like that. And the other thing, and I don't have any evidence for this at all, but just something fun that I was thinking is that maybe one of the reasons why the two-head pattern can be so compelling to a one-headed worm is specifically that it's different, not novelty. It's unique. It's like we've been a one-headed worm for 400 million years, and here's this new way of doing things, and most of us are not going to do it, but 14% of us are like, you know what, this looks interesting, right? And there's some explore exploit thing going on where there's like some small percentage looks at this and says, this is a crazy way to be a worm. Let's try that.
I love it. Great idea. Yeah.
I don't have any data for that, but it's a thought. Yeah.
Yeah, it does seem that a lot of these systems, when you add a stochastic element into the network, what it generates is not a random set of cells at the end of a lineage tree, but it generates a structured lineage that can have odd components to it. So this whole quasi-systematic, the diamond distributions, I had wondered about where the different head types you get in the planaria, and then a few odd forms that turn up occasionally are points in that diamond distribution, and whether you've actually mapped, if there is any way to map the space of what are the possible head types.
Mhm, mhm. Yeah, that's right. That's very important. And one of the things we're doing now is, I mean, some of the technology really needs to be improved, but we're trying to, with new advances in bioelectrical imaging, we're trying to get a much richer data set on the planarian bioelectrics, and specifically because we know the pattern that determines number of heads, but the shape of the head we really don't. And so now we finally have, I think, a way where we can get the resolution needed and try to really map out the space of yeah of possible heads and things that are much stranger that what else, because we've made things that, I mean, they don't even, they don't look anything like a planarian. They're not even flat. That's a whole other thing, is how you keep flat. But um yeah, I mean, another thing to talk about this, the lineage thing. So let me describe the following idea and see if it makes sense to you. um there's a I think there's a spectrum, and the spectrum is represented by C. elegans on one end and planaria on the other end. And what I think the spectrum fundamentally is how you deal with an unreliable medium. The thing with planaria, remember something interesting about planaria, unlike the rest of us, when we have offspring, they don't inherit our somatic mutations, right? The soma is disposable and all you regenerate from the egg. The planaria that we work with don't do that. They tear themselves in half and regenerate, which means that you inherit every mutation that doesn't kill the neoblast you inherited, and then it's going to be part of what produces the next set of tissues. So they're incredibly messy. They can be mixoploid. They can have different number of chromosomes. Basically, if you know from the start that your material is unreliable, then, and we've done simulations of this. So we have a couple of papers looking at simulations of what happens when the material is competent, meaning that it has certain homeostatic properties on its own, that the genome sets the hardware, but it has some problem solving ability anyway, and also unreliable because you never quite know what it's going to do. What we find is that evolution then exerts most of its effort on cranking on the algorithm. It stops, like the pressure. I mean, I think Steve Frank talks about how when we started having RAID arrays, the quality of each of the disk media went down because, right, the pressure is off. So something like this, the pressure goes off, and you end up with the animal that's the most regenerative, the cancer resistant, it doesn't age, the thing's immortal basically, and all of that with a really dirty genome. And it's the
Mm.
opposite. It's the opposite of everything you've ever learned in biology, where they tell you that you
Yeah.
keep your genome clean and all that. So I think what happens in C. elegans is that because of the hard coded, basically that strategy goes, we're going to do the same thing every single time. We know what our lineages are. We're not going to try to regenerate. We're not going to. There's no plasticity. We're just going to do the same thing, and that
Mm.
works perfectly well. And then what the other end of the strategy is, we can't rely on knowing anything about our substrate. We are going to be maximally improvisational. And even the developmental mode of planaria is often called chaotic because you don't know, unlike for other creatures, you don't know what the lineage relationships are going to be. They make it up as they go along for all of it. And they're
Mm.
very, very plastic. And then amphibians and humans are somewhere, or mammals are somewhere in between. We have a little bit of plasticity, but certainly
Yeah.
not as much as planaria. So I think it's really interesting to kind of think about the interactions between the uh, autonomy and this approach where you improvise. You don't go for fidelity and error correction and separation of layers and all the stuff we love to do in our computer architectures, but you assume your memories are unreliable, both your genetic memories and your behavioral memories, and all you're going to do is tell the best story you can at any given moment. And then it seems reasonable to me that you can get xenobots and anthrobots and all this crazy stuff, because I suspect that when you make a tadpole with an eye on its tail instead of in the head, they can see out of the box. Like, you don't need adaptation, no selection. No, it works fine.
It's amazing.
It's completely amazing.
Yeah.
Except if you accept that, I think that's because it never assumed it was going to be in the head. It works fine when it's in the head, but I don't think it ever assumed that at all. I think it tries to reconstruct some viable
Yeah.
story from scratch every time.
So thinking about some of Nick's simulations of C. elegans and the difference, because I was thinking, how would you repeat his simulations with
Mm.
Planaria? And it's crazy what would actually work. one question it raises is we had a very, very simple morphogen gradient in the C. elegans that Nick was doing. So the system itself, the genetics, because they can afford to be stable, the internal gene regulatory system, the expression network, the bioelectrics of it can be determined based on its developmental history.
Mm.
It pays a little bit of attention to the morphogen, but the morphogen gradient doesn't push it around very much. The morphogen gradient is a very weak operator in that system, and the curried function is very strong. Whereas my question was within Venaria is do they create reliable morphogen gradients, and are the morphogen gradients themselves actively sorting themselves? Because if you have autonomy in the gradients and the gradients are active elements, then you can afford to have very weak, you can afford to pay not nearly as much attention to your operators, and your operators need to have a different um, a different internal structure, but you can afford to put the reliability into the world because you can't trust your own internal genes.
Yeah. Yeah, yeah. I mean, they certainly do have um morphogen gradients, and people have modeled some self-organizing and stabilizing properties of these gradients. I mean, what we've seen is that the gradients are downstream of the bioelectrics, because if you change the bioelectrics, the gradients will, will
Yep.
snap into line. Also interesting that if you try to manipulate them at the level of the gradients, you can control head tail. You can make extra heads, but the extra heads aren't scaled appropriately. In other words, they are, you've acted somewhere down on the hierarchy where, yeah, you get the tissue identity, all right, but you didn't get the scaling, circuits. Whereas if you do it bioelectrically, you get both are downstream of that somehow. Um, yeah, but I mean, that's another thing, is like looking at these gene expression cascades. Xenobots have several hundred they have a transcriptome that's different by several hundred genes differently than what they would be doing in the embryo. Anthrobots have 9,000, over 9,000, so like half the genome different transcript, and a very specific, they turn on a few embryonic things, but no Yamanaka factors, for example, no reprogramming kinds of things. And yeah, it's just amazingly interesting how they choose which things are gonna, people say environment. I mean, the environment is extremely simple. They're in a bowl of cell culture medium, just like they would have been in the body, except that they're not next to other cells. That's the difference, is that they're not being influenced by other cells to be a nice trachea or whatever.
So I was wondering if you think about the bioelectrics as being a very, from a computational perspective, they're very fluid. They can be multi-scale. They can integrate across an entire organism. Um, they can also, they also don't have to reside within a given cell.
Yeah.
You can actually communicate the voltage to another cell, and then it can
Yeah.
take over a position in a gradient, for example. So they could do something that it would be harder to do with genes, which is they can switch the state of the cell very rapidly. Two cells could swap their states
Oh, yeah.
bioelectrically in a way that would be really hard to do genetically.
We see patterns move across tissues. We see them move,
Right.
and in fact we see them move across embryos. So in the latest work, we've actually looked at groups of embryos sitting in the same dish, and they actually will move across. They don't even obey the boundaries of the actual embryo. They will move across because the embryos are talking to each other. A group of embryos resist teratogens way better than, well, a big group better than a small group, better than a singleton. So
I, I think that's quite remarkable. That's a fascinating thing. And
mm,
I wonder whether that would also work, just thinking about some of the lingodroid studies, would that work across species as well?
mhm.
Could you could your patterns move if you have you think about the gut biome,
Yeah.
um, does it work across cooperative species?
Yeah. we're doing that now. So we had a paper showing that various bacteria that live on the planaria, they have a say in the shape of the planarian. So if you muck with the percentages of species, you can get planaria with weird visual systems and whatever. Like the bacteria make a difference, and we now have a project where we have xenobots talking to bacteria, and we're expanding this outward to see what are the what are the weirdest things that we can get to talk to each other. So
That's fantastic. I realize we're probably close to time. I'm not sure how much time you have, but I did want to ask, um, how do you communicate with a liver? So just communicating across species. Um,
yeah, yeah, right. So this is something that we're spending some considerable effort on this idea that we want to be able to talk to these talk to the body organs. And the approach we're taking now is we'll have something written on it in a little bit, but basically there's two ways you could imagine doing it. One way is to basically try to crack the code. So in other words, um, record bioelectrically. It will record everything you can under different conditions and see if you can work out, and it's some sort of machine learning or something. See if you can work it out. There's another approach which we're taking, which is to communicate to the molecular pathways inside the liver, and that is to use AI. And I'll show you. It's going to get dark here, so I got to go. But I'll show you at some point some of what we've done, trying to have a system that looks at a molecular network, at a real biological molecular network, and tries to put human understandable names on the nodes and the states, such that you can then actually basically to our goal is to have this being done in a human language, so that a normal person can instead of hey Siri, it'll be like hey liver, why do I not feel good today, and it'll tell you. So that's the idea. And so various kinds of AIs to try and make it understandable what it's doing, which is it sounds very artificial, but that's also, there's a model of the brain that's that, right? That basically one model is that you're just a big network that is good at doing things, plus a language model sitting on top that tries to come up with, confabulate some explanations for what it is that you just did and why. So at the very least, whether that's true or that, I don't know, but at the very least, I think we could construct something like that.
so one reason I'm really interested in how you're doing it and the processes are because of mentioned when we last met about the fungi project where we're trying to communicate with mycelium.
Mm, mm.
And so we would love to hook an AI up to the mycelium, and we're currently at the stage of just doing a lot of recordings and trying to decode
Yeah,
what signals mean in different cases. But I think it's really interesting trying to learn from how you say good morning liver
yeah,
to good
yeah.
morning mycelium.
I think that'd be great. Have you that'd be great. Have you talked to Olaf Witkowski at all?
No.
He's got some, so maybe at some point I'll set up a conversation with you, me and him, and we can bring our people, we can all talk about. He's also talking to some yeasts, I think, is or bacteria, and I think he has a different yet approach from what we're doing. And so I'll bring Yanbo, who's the guy in my lab who's doing it, and we can all kind of compare notes. I think that would be great fun.
That sounds great.
Yeah,
Yeah.
cool. Okay, well, thank you so much. This was incredibly interesting. A lot of amazing work. So yeah.
Oh, thank you. It's a great pleasure to talk to you. Your work has really made me rethink a lot of the fundamentals, and I'm fascinated